Hello students, this is Dr. Fazan Mirza. I am discussing with you the light independent reaction which occurs through Calvin cycle. We know that the Calvin cycle occurs in the stroma of the chloroplast, and how it occurs, we know it carbon dioxide fixed into sugars, it used the reducing power of NADP and ATP as well from the light dependent stage. So carbon dioxide enters into the stroma. In the stroma, it is turned into 6-carbon compound, which is an unstable intermediate. This 6-carbon compound found because carbon dioxide having one carbon, it combined with a compound called RUBP. RUBP stands for ribulose bisphosphate. Ribulose bisphosphate comes from a sugar called ribulose. So ribulose is a sugar having bisphosphate means 2 phosphates. So ribulose bisphosphate is a 5-carbon compound. This 5-carbon compound combines with carbon dioxide, which is having one carbon, and forms a 6-carbon unstable intermediate. So this is how the Calvin cycle begins. The 6-carbon compound is an unstable compound, so it quickly just breaks into 2 molecules of GP. The 2 molecules of GP are having 3 carbon each. The 2 molecules of GP then turn into 2 molecules of TP, and TP is 3 carbon again. But this reaction uses the reduced NADP to turn into oxidized NADP. This is where the NADP is getting oxidized back, and then this NADP will go back to the light dependence reaction to bring more reduced NADPs or more hydrogens to this reaction. NADP is broken down to ADP as well. So yes, oxidation of reduced NADP, reduced NADP and the hydroxy of ATP occurs here. The two molecules of TP can then be further converted to ribulose bisphosphate using more ATP from the chloroplast resources, whereas a major amount of triphosphate can be used to convert, can be added together to form glucose with 6-carbon amino acids and lipids and any other compound that the chloroplast would need. I have added the fractions here to tell you that how much triphosphate is being converted into the products as food and how much of it is being used to regenerate RUBP. So you can see the fraction that 1/6, which is a smaller fraction. I think I said it otherwise in the other way around previously. So actually a smaller fraction, which is just 1/6 of the total triphosphate, it is used to produce these compounds, whereas 5/6, which is a larger fraction, it is used to turn this back into ribulose bisphosphate, so Calvin cycle can continue. The same TP can be, which is just a smaller fraction, can be used to make any of these compounds which the chloroplast would need. These plants are called C3 plants because the first stable compound which is made in these plants is a 3-carbon compound, which is GP. RUBP stands for ribulose bisphosphate, as already I've mentioned with you. What about GP? GP is glycerate 3-phosphate. And what about TP? TP is triose phosphate. So this is how these chemicals work. And then there's an enzyme Robisco. Robisco is the most abundant enzyme on earth. It works on pH 8. This pH 8 is present in the stroma, and this Robisco keeps combining the RUBP to carbon dioxide. They get added together, so the Calvin cycle just can continue. So this carbon dioxide we know is acidic in nature. So still Robisco maintains the pH of 8 in the stroma because carbon dioxide is not allowed to accumulate. All the carbon dioxide that comes, it quickly get used up in the Calvin cycle inside the chloroplast. So the Calvin cycle, as it proceeds, carbon dioxide is not allowed to accumulate. For example, if I take this question here, you can see that the light was turned on at this point. GP concentration was stagnant and RUBP was stagnant as well. But as the light was turned off, GP started to rise and RUBP started to fall. What's the reason? Why GP got higher and RUBP got lower? Basically, GP increases as the light source is turned off. Why was that so? We see that RUBP will keep on getting converted ahead into GP. As Robisco keeps combining more and more carbon dioxide and RUBP together, this goes on and on until all available RUBP is used up to turn into GP. You can just refer to the diagram that I already have discussed with you in this here. So you can see that all RUBP will keep on turning into GP, and since GP is not turning into TP, the amount of GP will keep increasing. And that's exactly what you see here in the graph. Until all GP is being used, all the RUBP is being used up. What about RUBP? RUBP, you can see its concentration just drops down. Its concentration drops down because as the light source is turned off, light dependent reaction stops, and when the light dependent reaction stops, this stops the production of ATP and reduced NADP. And we know ATP and reduced NADP are mandatory for converting GP into TP. And since GP is not converted to TP, so GP keeps increasing. Yes, so it keeps increasing again, as already discussed. So yes, it keeps increasing, and when GP is not converted to TP, RUBP is not regenerated either. So when it's not regenerated either, RUBB quickly just declines upon being converted into GP as the light source is turned off. So you can just go back to the diagram that we've discussed for Calvin cycle and see how this relates to this particular stage in which GP is turned into TP and the products of light dependent reaction are being used up. Limiting factor is the last thing that we discussed. Limiting factor actually tells you how much amount of which factors actually tell you how the rate of forces will increase. So for example, light intensity is one of the major limiting factor. Apart from this, carbon dioxide concentration is also a major limiting factor. A limiting factor is a factor that limits the rate of reaction. So any variable that I'm taking on the x-axis, for example, I'm taking light intensity, so it will be my limiting factor. But it will be my limiting factor for rate of photosynthesis until the rate of photosynthesis keep increasing. So if I am increasing the light intensity and the rate of photosynthesis keep increasing, so from point O till point A, yes, light intensity is the limiting factor. But beyond point A, any point on this graph, if I ask you what's the limiting factor, light intensity cannot be the limiting factor because even increasing light intensity doesn't increase the rate of photosynthesis further. Similarly, if I repeat the same experiment at a high carbon dioxide concentration, I see a huge jump. So this graph became stagnant, or it became plateaued or parallel to the x-axis because carbon dioxide became the limiting factor here. So let me repeat this experiment. If I was able to repeat this experiment at higher carbon dioxide concentration, what I would see, the graph is way more steep now. Yes, between region O and B, the limiting factor is still light intensity because as I increase the light intensity, the rate of photosynthesis keep increasing. So between O and B, any point here, the light intensity will be the limiting factor. But beyond point B until point X, since the graph is becoming parallel to the x-axis, whatever is mentioned on the x-axis cannot be the limiting factor. So what will be the limiting factor here? The limiting factor will be again carbon dioxide concentration. Similarly, temperature can be a limiting factor as well. Water availability can be a limiting factor. The number of chloroplasts, and there are many others as well. So yeah, the major ones are just carbon dioxide concentration and light density, and to an extent temperature as well. How's this important for this information is important for commercial plant growers, because these commercial plant growers can manipulate these factors to maximize the rate of photosynthesis and therefore increase their yields of their crops. What they can do, they grow crops in glass houses where they can control the light intensity, temperature, and the carbon dioxide concentration artificially. They can cover the crops growing in an open field with a transparent plastic bag. For example, polytunnels. This increases the temperature, and as the temperature gets increased, we know that the rate of photosynthesis will increase as well. They can irrigate crops in open fields, and when they do that, they are providing a lot of water. And again, this will just keep on making sure that the rate of photosynthesis keeps higher as well. So yeah, that's it from my side. Thank you so much.